Okay. Let's get the show on the road, shall we? So, that look familiar? We finished 10 last time, and we are getting a clean start on 11. So let's go ahead and get started. First of all, 11 will probably go quickly because there's a couple of things, I'd say maybe 2, 3 things that are like really different. A lot of overlap in 11 with mitosis, right? So when you're talking about, the basic phases of mitosis, the phase of cell cycle, like prophase, metaphase, anaphase, telophase, a lot of those are recapitulated here in meiosis as well. So there is a lot of that heavy lifting that we've already done, right? So 11 tends to go pretty quickly. And not only that, but typically use chapter 11 rather than just studying meiosis as its own, and I would argue beautiful, cell division mechanism itself. I oftentimes see 11 as sort of like, it's kind of like pre-calculus, right? You're kind of preparing for calculus. You're getting yourself ready. Of course, in pre-calc, if you're taking pre-calc, you know that pre-calc is basically just a fancy name for trig for the most part. But you're basically learning some of the principles and sort of the things, the mechanistic issues that underlie calculus, right? So the next big leap, and that's kind of where we're going. So we see meiosis as not so much as sort of like, oh, well, here's another type of cell division, as much as this is the mechanism for genetics. So in order to understand 12, we need to do a little bit of front work on the mechanism of genetics, which is 11, right? So that's kind of that's the way I approach it. So let's approach it that way, right? So this is the sexual reproduction, right? And meiosis chapter. We're finally here, right? So for us multicellular organisms, we're finally at a reproduction chapter. It took 11 chapters to get there, but we're here now. So let's take a look at this. First of all, I want to compare our 2 major reproductive pathways, right? So in the last chapter, we looked at cellular reproduction. So how does a cell reproduce itself? You go from 1 cell to 2 cells. Now we're talking about organismal reproduction. And there's really 2 main ways that you can do this. And I want to kind of sketch this out a little bit and do a little bit of audience spitballing. We're going to basically sort of take a look at this a little bit, because what I want to do is I want to sort of get this off on the right foot. There's 2 dominant ways to reproduce. One is sexual. However, the other one is asexual reproduction. Now, if you take a look at what bacteria did, right? They just basically divided and split, and 1 became 2. That's a type of asexual reproduction, right? Anytime you're making a clone of yourself, that's a type of asexual reproduction, right? So essentially, when you take a look at asexual reproduction, what you're basically doing is making a photocopy of yourself genetically. Just imagine how frightening that would be, right? The only thing worse than a world with me in it is a world with 2 of me in it, right? So let's take a look at this. The pros and cons of each. This is what I want us to sort of spitball out, because I want us to understand very clearly why there's a preference for one and not the other. Okay, so let's think about this. Okay, first of all, I'm assuming that when it comes to sexual reproduction, we're all pretty good, right? We've already had our birds and the bees talk. We're old enough to know a few things, right? So we've been there. So we kind of have an idea of how that works. This is not a human sexuality class. We're not going to get like deep in that rabbit hole. So but we basically have a pretty good idea of at least the requirements, right, to pull this off. Asexual, on the other hand, is a little bit different, right? And so we've seen some asexual reproducing things. Bacteria is probably the most common one, right? So essentially, you just make a carbon copy of yourself. So here's the thing I want you guys to do, and this is audience participation. I just want you to start tossing some stuff out there, and we'll kind of put it out here and try to sort it all out and figure out where it all goes. So you can start anywhere you want, either for asexual or sexual. Give me a pro. Answer this question. Why do it this way?
I would think a pro would be only one individual to do it.
Yep. Mm-hmm. So then the converse of that would work as a con for sexual, right? That basically means you got to talk somebody into it. Okay. But wait a second. There's just an obvious efficiency associated with it, right? I mean, anything you can do on your own is always more efficient than something that you have to rely on somebody else to do. But wait. Let's actually blow this up. Let's think about this for a second. Let's scale this. When you're talking about reproduction, think about this for a second. What does it take to talk somebody? Yes. Okay, hang on to that one. Hold it. But what does it take to talk somebody into it? Is it an easy thing? You just call somebody up out of the, just randomly, just cold dial somebody. Hey, you want to mate? By the way, don't do that. That'll be like guaranteed pepper spray moment right there. Right? I mean, is that how it works? What goes into that? Events.
Is what you're looking for.
Okay, so it depends on what you're looking for. Go. What are we looking for? What? The colorful feathers, right?
Very nice.
Good, right? Got to have the 6-pack, right? Got to have something to look at. Think about it. You're thinking like a mammal. This is what I love about this particular discussion, right? Because we can't help ourselves but think like mammals. And this is it, right? Okay, so wait. So what about the dude who comes along and he's got sort of dull looking feathers and it's all monochromatic? Is that the one you're signing up for?
No, let's face it, girls. Come on. I mean, come on.
Be honest. The guys aren't off the hook either, right? I mean, there's a reason why marketing works, right? Because we're mammals and we can't help it. Right. You go for the colorful feathers. But what about, wait. What if this guy's colors are just a little bit brighter than this guy's? Who's going to win that one? The brighter guy, let's face it. Come on. I mean, honestly, right? I mean, be honest, right? Think about this. Okay, so this is great. This works great if you're a peacock, right? We get to see all animals out there. The dudes are kind of strutting their thing. They're kind of like flipping their little feathers around. And of course, the peahens are just like, right? And and that's all animals are doing that, right? But think about how much goes into that. How much biology goes into that moment when the peahen makes the decision, I want you. Oh, really? Why? Why did you pick him and not him? Right? There's an entire biology behind that. And we call that mate selection. You hear a lot about natural selection and like evolution. That's basically, does the predator pick you to eat, right? But mate selection is also a very potent biological mechanism. That's basically, how does one particular individual select a mate? And by what criteria? And how do they formulate the rejection, right? So there's a lot that goes into that. By the way, if you want to have any kind of, and some of you guys already know this because you're already there. If you want to have any clue just how big of a deal this is, we've got entire industries based on this, right? Like Match.com, right? Any of those dating websites, those find your soulmate websites. I don't know how many there are. I lost count around a dozen, right? And that's before you get to like just the hook up and shack down sites, right? Which are just like, listen, I don't want to thing with you. I just want to just like animal like mate with you. That's it. Just like basically it's online prostitution essentially. Right. That's kind of what it is. But that's all part of this, right? That's all part of this. You got to talk somebody else into doing it. And there's an entire process to that. And there's a lot. Now, this is where you come in, Megan. And that requires a lot of energy. That already is a bad mark against you, right? Well, how about asexual energy-wise? This is much more energetically efficient, isn't it? I don't need all those websites. I don't need anything. I don't need to go through all of this ridiculousness of mate selection. I don't need for somebody else to grow big, colorful feathers. That's save your money because I'm asexual. So I don't really need anything else. Think about how much energy that would save. All these organisms that are out there, the peacocks are a perfect example. By the way, birds are a great example because they're heavily sexually dimorphic. Almost always, who's the colorful one? The males. Why? For this reason. Because the dude is trying to look good for the girl. That's what he's trying to do, right? Why do the girls always look kind of drab and brown? Not all birds, but many species of birds, because the hen will lay the egg and rear the chicks, at least until they're able to fly. So the last thing you want to do is for some brightly colored female bird trying to protect her defenseless little babies to basically have a big old sign on her head of color that says, here, eat me, you can see me, right? So a lot of times the female birds are more camouflaged because of that for predator avoidance. So who are they going to eat first? The guy, right? That's the one that get eaten. Now, not all birds are that way. Some birds are not sexually dimorphic. Crows, for instance, are not sexually dimorphic, but many of them are. Okay. I don't know about you guys, but right now, the list we got going on seems to be leaning kind of heavy in favor of asexual. So can you think of any pros for sexual? Yes, I love you, Megan. That is it. Variation and diversity, right? So now what does that mean about asexual? Yeah, you have almost no diversity, right? So there's no variation. How big of a problem is that? I mean, is that just an annoyance or is that a significant problem? Yeah, variation is how you evolve, right? Think about it. So these are some nice ones. You guys pretty much hit on, or you guys are really good, efficient, very efficient. You pretty much hit on all the ones that I usually take a lot of time trying to tease out. But think about it, right? Most of the world that we see is sexual. Very little percentage-wise of the overall biological organisms are asexual. So if asexual is so energetically efficient and so like, straight to it, then why would you do this? Well, what that means is this idea of having no genetic variation must be a catastrophic fail for asexual, and it is. Think about this for a second. The only reason why I do asexual reproduction is for one reason and one reason only. If I've got a genetic variation profile that essentially fits to my environment perfectly, that is to say that my genetic information allows me to survive in my environment perfectly, then there's really no need to change, right? However, is the environment unchanging? It changes constantly. So if the environment changes, then as an asexual organism, what are my choices? Yeah, die or change. That's literally it, right? But how do I change? I have to go by the slow grind of mutation and just hope and pray, right? But we know enough about mutation rates that if that is where you're putting your money, you are a fool, because it takes like a long, long time for a mutation, first of all, to actually yield fruit. Right, so like a beneficial mutation. Mutation rates that actually give you variation that allows you to be able to adapt to your environment can be grindingly slow. Like maybe 1 or 2 per, I don't know, couple thousand meters, perhaps. That's pretty quick, actually. Think about it. Your environment's changing now. What is the likelihood that randomly, because that's what mutation does, it's random change, that you're going to get lucky and you're going to hit the lottery? It's like, maybe I just might get that random mutation that could save my life. Is that a winning strategy? So most likely then, if your environment changes on you, what's going to happen to you very essentially? You're going to die. Okay. However, if you're sexually reproducing, then you've got a lot of genetic variation. And the name of the game, the purpose, the raison d'etre, if you will, is variation. So a lot of times students will ask, well, wait a minute, why? Just why, right? Why are we studying meiosis? Why is meiosis a thing, right? Why does it even exist? What's the point? I mean, don't we already have mitosis? Why do we need another one, right? Why do we need to do this at all? The answer to that question always comes back to what? Variation. Basically, when you look at meiosis is like a random number generator that constantly mixes up your outcomes so that you never get the same outcome twice. The difference between asexual and sexual reproduction, I always like to sort of do by analogy, one that really hits home. Let's say I give you guys an exam, and I'm going to start it off with. So it's an exam, but the good thing is, I'm going to give Connor the exam, but I'm also going to give him the key. So what does he actually do? Just fills out the key, right? Of course, you get 100 on it. Life is good. And then what happens? Connor basically reproduces, right? And he sends the information that he has, which was a perfect match for his environment, right? The answers that he has, the key, is a perfect alignment to his environment, which is the exam. So if I then move the exam forward, right? Do you change anything? No, because you want to pass the exam too, right? So what do? You just take the same instructions that Connor had, and basically you just fill out the key. Why? Because the answers fit the exam perfectly. So then when you pass it on, are there any changes? No, you just continue to fill out the key as long as you go through the entire process, right? This is a perfect example of the answers that you have match your environment perfectly. And we have success. So why mess with it? All you do is duplicate success. But that works, and you'd all get A's. You'd all be very, very happy. But at the end of the semester, you wouldn't know a darn thing. Right? So the A would literally mean nothing. And then CCD would lose their accreditation, and then your degree here would be basically about as valuable as toilet paper. But about as valuable as toilet paper, COVID shutdown not included. For some reason, it became more valuable than money. But I never did make that connection. I mean, that just, to me, highlighted the stupidity of Americans. It's like, oh, we're getting shut down. Let's all get toilet paper. Like, what the hell is your problem? I mean, honestly, it's like serious. I mean, it's like beyond lemons. It's like retarded or something. I don't know what the problem is there. Anyway, if you, by the way, if you understand what happened with that, just explain it to me, because to me, there's a whole lot of stupid when I think about it, which is actually quite disturbing, actually. I have to admit, for the first time in my life, I was a little ashamed to actually be an American. The way we kind of handled that. I mean, you've won wars, you saved nations. Seriously, this is what you do now. You basically wet yourself over something like this. That's okay. I'll stop. But now let's do something different. We've got a plan for success, yes? Don't change it. This is where asexual reproduction will really, really do well for you. Okay, but wait a minute. Let's say I kind of send the exam through all of you guys, and then it comes to Mobina, and I like to pick on Mobina. But now what I'm going to do is I'm going to change the environment. I'm going to change out the exam. I'm going to give her another exam. She still has the key. How well do you think Mobina is going to do? Where all she has is the key. That's it. Oh, no cheating. You didn't study, right? Because this isn't a biology exam. This is like something that you guys, haven't even studied, like music theory or something like that. You musicians, no cheating, right? Imagine you're not musicians, right? Or something of that nature. History, if you're, that's your black hole, right? But that's what I did. So now you've got all these answers, all this information for an exam that I've just changed on you. Not only that, but guess what? You're not studying the subject, so you have nothing to fall back on. You can't think your way out of this one. All you have is your key and an exam. How well do you think Mobina is going to do? It's going to get ugly, isn't it? Right? And she has the same options as the asexual. Either change or die. Well, how does she change? She guesses and hopes that she guesses right. What are the chances that she's going to guess right enough times to do well in that exam? So there's a strong likelihood that she is basically going to fail. The exact same situation happens with asexual. Because mutation grinds along slowly. I mean, on a good day. So if you're basically saying, I'm going to mutate my way out of this mess, good luck to you. I'll send flowers to the gravestone, right? Because that basically means it tilts heavily toward dying. So think about that. We already know now that our environment is more chaotic than it is constant. So the chances of our environment being stable enough for asexual to actually work for us is probably very unlikely. Just like with your exam, right? The exams change enough so that you kind of can't just come in with that key, because it's always going to change, right? Because I'll change up the answers, I'll change up the options. So you can't actually rely on the key, even if you got one from last semester, because you know that the exam is not the same exam. I've changed it on you. So the only way that you can pass the exam is if you have some depth of information to bring to the exam. That's called studying. So why does Mobina do well on the different exam? It's because she was allowed to study, add variation, intellectual variation, which is knowledge, to her system. And it's from that pool of variation that she starts digging down into when she sees a question she doesn't understand. And like, well, wait a minute. So this I know, so I can dig down in here and I can grab this out. But guess what? It's got to be there in the first place, right? If you don't have that deep reservoir of knowledge, that variation of knowledge, then you don't have anything to dig down into in order to pull out that would allow you to be able to answer the question. Right? You guys do this all the time, right? How many of you guys live on process of elimination? We all do, right? So it's okay, right? But what are you doing in process of elimination? What you're doing is you're digging down into your knowledge variation. You're saying, well, I don't know the answer to this question, but I know some things that I actually aren't true, right? So I can eliminate some things. And so here's a little piece here that I can use to eliminate this one. I can use this little piece here to eliminate that one. And at least now I've got a fighting chance, right? That's your variation of knowledge. So when you're taking a look at a sexually reproducing organism, for you, that variation of knowledge is genetic variation. So when the environment changes, you don't have to mutate. All you have to do is reach down into your genetic variation, which is already there, and just dig out, hopefully, the right solution to the problem. That's the reason why this is so important, because the environment constantly changes, and we have to have as much information as possible so that we can adapt to that environment. Okay. So that's the reason why. And by the way, if you're wondering, I mean, obviously, meiosis is the story of how we generate that variation. Genetics is the story of how we pass that variation on to the next generation in response and in adaptation to our changing environment. So let's go ahead and take a look at it, right? So most of us are going to be sexually reproducing. And so we'll be getting genetics or genes from two individuals, right? So from mom and dad. And since all of our moms and dads are different, then we are basically getting our genetics from two different individuals, right? And so this is kind of creates our line of descent, our lineage, if you will. And ultimately, taken over multiple generations and over quite long periods of time, will relate ultimately in species change, which is evolution. Now, evolution, remember I said back in the beginning, evolution is a genetic thing, right? It's not a paleontological thing. Paleontology is basically a dead zoo. That's, by the way, if you're taking geology with Professor Adams, I love the guy to death. Don't tell him I said that paleontology was a dead zoo. If you're taking historic geology, that might be a little on the hurtful side, but it's true. I just don't want to offend him. And he's just nice. If you know him, Aaron Adams, he's a great guy. But it is, right? Because evolution is defined as basically the speciation or the change in species over time, right? So what is it that changes a species? It's their genes. It's their traits that change over time. So fundamentally, evolution is and always has been a genetic story. It's just, we're just now for the first time able to have the genetic tools available to us to be able to actually recast evolution within its correct lens. And I would argue that having it sort of not in an evolutionary lens created more confusion and conflict really than anything else. Because it is what it is. And there's a lot of nuance there. I'm not going to get into this, right? Because I mean, obviously, there's a lot going on, and just kind of following these changes in genetics over time. There's so much going on there, and there's so much depth there, that it really is something like you have to be able to just really focus on it and do it for like an entire semester. It's not really, there's certain things that you in life where it's like either either do it right or don't do it at all, kind of a thing. Evolution is very much like that. To kind of sock puppet our way through it, which is oftentimes what general bio books try to do, is really kind of an injustice. And it really a lot of times casts a lot more confusion and a lot more inaccuracy on how people see evolution, because you don't see it like fully decompressed in its full glory, right? And that's the reason why I tend not to talk a lot about it, is not because it's not fascinating, but because unless I have the time to really decompress it and pull it all out so we can all see the different sides of it, it's honestly not worth talking about it at all, right? Because it really is something that requires us to have our full mind and attention and focus engaged on it. When you start to sort of shorten it up into sound bites, it becomes honestly, it becomes a little artificial, to be quite honest with you. It comes across more like, just trust me. And that is the ultimate non-scientific thing to do. It's like, why, right? Why should I trust what you say? So and I don't expect anybody to do that. And I don't want to put people into that position to say, just trust me, right? Like pretty much everything I say is crazy. I'll tell you full out if it's my opinion. But I'll also tell you, it's like, no, this is one we've documented and I can actually find the research papers where we actually pin this one down. I think that's important for instructors never to get too far away from that. Because I don't think it's fair. Because a lot of instructors did that to me and I didn't feel like that was fair. To basically say, oh, listen, I'm the instructor, so you just trust anything I say. I could say any crazy, wild-eyed, nutsy, cuckoo thing in the world, and you have to just believe me. It's like, well, why? Why, right? I mean, scientifically, this is kind of, you're training us to be critical thinking. That was one of the first conversations we started off with in the semester, right? Think critically, question everything. Question me, right? Because when you guys question me and you kind of put me back on my heels a little bit, well, I love that, by the way. Then it kind of forces me back into like, you got a good question. Let me go back and sort of refresh my memory where I got that from, right? One of the most recent examples of that I did was remember the archaeology thing about the gates and the gate complex thing? Well, one of the things was and nobody really challenged me on this one. I just sort of challenged myself. It's like, after a few years, you want to make sure that the way you're presenting it is as accurate as it originally came out, that you don't start falling prey to sort of like the fisherman's, tales, and you want to make sure that it's scaled correctly. And I recently went back and I searched out all of the history of that discovery in archaeology just to make sure it's like, okay, yes, the way I'm describing it is the way that it was discovered. So it's an accurate representation. On that side of things. So that's an important sort of a thing, right? That's an example of me making sure it's like, well, I have to make sure that, I'm just not up here, just saying Lord knows whatever, and just somehow you have to believe me because I've got your grade, whatever. I mean, that's just stupid, right? I've had a lot of professors do that. And I'm naturally curious. So I asked lots of questions and they would get all like, oh, dare you question me? It's like, well, why wouldn't I question you? I mean, you're training me to be a researcher, which means you're training me to ask questions, right? So why wouldn't I ask questions of you, right? I especially zero in on that, and I want you guys to zero on that too. Whenever you hear somebody lay down a blanket dogmatic statement, right? Like if they say, oh, well, this is the way it happened. Really? Well, wait, question. So does that mean you're 3.5 billion years old? I'm thinking of like the endosymbiosis theory, right? The one where the eukaryotic cell gobbled up the little prokaryotes to become like mitochondria and chloroplast, right? That's a perfect example. And that's the reason why I say it's like, I want you guys to keep me honest and keep your professors honest, right? Because that's the reason why I don't like to get too deep into evolution, because it forces you, if you don't have enough time to unpack it, to sort of do those sorts of things. It's like, well, this is the way it is, and this is the way it went. It's like, okay, but how do you know that? Right? How do you know that? Because we can see with the endosymbiosis theory, I could have picked any one of those and said, this is the way it is. No, not really. Even your own colleagues don't agree with you because there's some people who maybe favor some of the other half dozen potential explanations for endosymbiosis theory, right? And so they would disagree with you. So who's wrong here? You or them, right? And that's a terrible place to be as a student when you're sort of saying, well, wait a minute, you can't be right and these guys be right at the same time, right? So what's going on here? Who do I believe? Right? That's a terrible place to be. That's the reason why I always want you guys to always push back on me. If you hear me say something that's a little, over the top, I'm usually doing one of two things. I'm either hyperbolizing something for the purpose of you remembering it. That's a thing, right? Or I'm blanket just stating an opinion, in which case I'll admit it's an opinion, but I'll also own it. And usually I won't back away from it because we all have a right to our opinions, but they are, after all, opinions, right? That's the reason why when I try to talk about evolution, I like to talk about all of the potential angles, the competing ideas, not like this is the way that it was, right? Because then it starts sounding like Genesis, right? So in the beginning, blah, 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 blah. It's like, no, that's not the way science works. Science says, listen, this is our idea. We have like multiple hypotheses around this one. We need to sort of vet each one of them and sort of just take the pros and cons of each and sort of figure out where we land, right? That's the way science works. And that's the reason why sometimes I won't talk about certain things. So a lot of times students will ask me, but at this point in the semester, well, how come you don't really talk about evolution so much? That's the reason why, right? Because unless we have the chance to really unpack it, we're not really doing much with it. Okay. So let's take a look at our meiosis. So we've already talked about our diploid cells, right? So these are the ones that have our two sets of chromosomes. This is true for all of our body cells, right? So our somatic cells are basically our body cells. And so we've kind of looked a bit at this a little bit, right? So the whole diploid, haploid idea. So we kind of broke this down a little bit. So I don't want to sort of belabor this one too much because we've already been there. And then, of course, we have our haploid cells, which are our gametes. So our gametes basically are the sex cells that we have. So this is our eggs and sperm in us. And so in this particular case, we only have one set of chromosomes, right? So all of our unique chromosomes. And so when we take a look at the offspring after fertilization, we have one sperm with 23 chromosomes, that's our haploid number, plus one egg with 23 chromosomes, goes through fertilization, where the sperm fuses with the egg, and then the male pronucleus will fuse with the female pronucleus, and that will constitute the new zygotic genome. Because now we have one cell with 46, right? So this is the zygote. And this is where we start off. This is basically where we start at the top of that mitotic tree when we start to build our body, right? So we all start off with as a single cell, and then we basically go through multiple rounds of division to build our body. So this is it. So you can see here, you've got your haploid sperm, which is carrying half of the father's genome. And of course, your haploid egg, which is carrying half of the mother's genome. They fuse together, and then you get your diploid zygote. By the way, if you're keeping track, you just got another version of the birds and bees talk. Right, that's basically it. You'll have to wait for anatomy and physiology before you get to the hardware portion of the birds and bees talk, right? But this is basically kind of the basic mechanistic component of it. Okay, so let's take a look at this then. Now, we have these diploid cells. That's most of our body, right? So what you see in the mirror is a diploid structure. So all of your body cells have these two sets of chromosomes. And the only ones that have the haploid set are your germ cells, right? Which are busy basically going through meiosis to create your gametes themselves. Now, generally speaking, animals as a group, the entire kingdom is largely diploid dominated. That basically means that your body, what you think of as your body, is dominated by the diploid cells, Okay, that's kind of like us. This looks like us. However, the same thing is not the case for other organisms. Some organisms will actually have a haploid body, and then they will toggle between a diploid body and a haploid body. This is what's referred to as the alternation of generations. So obviously we'll be spending most of our time on our situation where we have the zygote going through mitosis, producing our body that is full of diploid cells. And then ultimately we will have a subset of these diploid cells, which will then go through meiosis to create our gametes. And then these gametes will become the sex cells that we use for fertilization for the production of the future generation. So this is what our life cycle looks like. So upon fertilization, let's start off with the zygote. We are diploid, right? 2F. We're going to be going through mitosis, multiple rounds of mitosis to create our body when we're born. We still go through mitosis until we become reproductively mature at adulthood. Notice we are still diploid in body because all we've done is mitosis. Remember, mitosis just copies what already exists. However, in the ovaries of females, a subset of those diploid cells will then go through meiosis. Same thing with the testes. In males, subset of those cells will go through meiosis. And when they do, they will generate the haploid sperm. Now notice your sex cells are haploid, and in the females, you produce the haploid egg, which will then fuse quickly to essentially create the next generation. So notice, our existence in haploid state is a very short period of time. So if you don't have any sperm, you lose the egg. You start all over. Okay, that's what happens during menstruus. Essentially, the thickened uterine lining, which thickens for the potential implantation of a fertilized embryo, realizes that there is no fertilized embryo to implant, and so therefore you let go of that upper layer of thickened uterine lining. So you lose that egg. That's gone. And that doesn't take very long. The sperm, same thing. It's use it or lose it. Right. So if there is no fertilization, the male roughly is making a new batch of sperm roughly every 24 hours ish or so, a full batch. It's kind of a continuous process, but he'll make a full batch roughly every 24 hours. You either use it or you lose it, but it's a very short-lived period of time. So this haploid state is very, very short. Most of our life is dominated by the diploid state. So that's not the case with some organisms. So for instance, in some organisms, just to show you what this alternation of generation, I just want to do this just to kind of foil this so you can kind of see that not all organisms are the same. Let's take a look at something like mosses. So moss is an interesting little thing. It's not quite plant. It's not really animal. It's a very kind of primitive in the plant kingdom area, right? It's kind of it's like in the bryophyte area. So it's kind of like what we see as sort of like a primitive plant, right? So like before gymnosperms, which are like the cone-bearing plants, like pine trees and things like that, and then before angiosperms, which are the flowering plants, like back before you really got these, like what we recognize as a plant, where the plant sort of grows out in these like little embryonic leaves, and then it gets its first full adult leaves, and it grows into a stem with leaves and whatever. Right before we get to that one, the whole plant world was very crude, and so all the structures are very crude, and mosses are seen as sort of like that early sort of version of what kind of plants became. But basically, plants have two different bodies to it. So let's kind of show you what they are. The first body of the mosses, is a diploid body. Called a sporophyte. So what does a sporophyte do? Well, the sporophyte will basically create what's called a sporangium, which is kind of like this long stalk-like structure, still diploid cells. It's got like a little pod on it. And inside, a group of cells goes through meiosis, producing little haploid spores. That's the reason why it's called a sporophyte. And eventually what's going to happen is that little sporangium is going to pop open, and those little spores will land somewhere. So this will be your little haploid spore. I'll draw it over here. And then the spore then goes through mitosis. And as it goes through mitosis, it creates a haploid body called a gametophyte. So here you have a haploid body that's called a gametophyte. And then what happens here is a subset of cells of the gametophyte will go through mitosis to create the haploid gamete, which will then fuse with the gamete of another individual, and that'll create your little diploid zygote, which will then go through mitosis again to create the sporophyte. Okay, that seems a little crazy, doesn't it? Probably lost you guys a couple of dashes ago. So it's similar to this, right? The only difference is what's happening here is you have your zygote, which goes through mitosis, just like us, right? It creates a diploid body, just like us. So that's your sporophyte. But it has a subset of cells that goes through meiosis, just like us, that produces, instead of gametes, it produces spores, which are haploid. This is where it changes. Unlike us, these spores will then go through mitosis and create the haploid body, the gametophyte. And the gametophyte is the one that produces the gametes by mitosis, right? Because you're already haploid. So you don't need to go through another round of meiosis. You're already there, right? And then it's these that get fused together to form the zygote. This is what it would be like in us. What happens if we did this, like mosses did? If we did this, what happened is when we had our mature body, our sex cells would produce haploid cells, sperm and egg. But now, instead of sperm and egg fusing together, which is what they do now to form the zygote, if we did what mosses do, what would happen is the sperm would actually start dividing, and the egg would start dividing, and then it would divide into a full body. So like the sperm would become like a half clone of the dad, and the egg would become like a half clone of the mom. So there'd like be an entire group of like haploid people walking around, which were like carrying half of the genetic content of their parents. Not only that, but what they would do is they would mitotically produce the gametes between each other that would then fuse together to make the zygote. And that would be the sexual reproduction. Wow, that's crazy. Right? So what that means is it's not the parents that have the kids. It's the kids that have the kids. So like you wouldn't actually have your sexually reproduced individual in the first generation. It's like your grandkids would be the sexually reproduced individuals. What you would have is a bunch of half clones running around. Now, how many of you guys does that sound completely crazy? right? You know, there's a science fiction novel out there somewhere doing exactly that, right? So what might be the benefit of that? Well, that actually could be quite beneficial, actually. Like, for instance, if you needed to sort of get away from your environment and you can't, like, say, for instance, mosses, they can't just get up and run around. This might be actually a pretty clever way to sort of spread yourself around and get yourself into more favorable environments. You know, it's kind of a spin-off, if you will, of that classic thing that we oftentimes will do, where I will say things like, you know what, I want to have a family, but not right now. Right? I want to work a little bit. I want to set up my career. I want to have a stable career. I want to make some money. And then I want to be able, then I'll have kids, right? So I want to make sure I improve my environment so that my kids have more than if I would have kids as a dead broke students in college, right? That's this is a version of that idea, that basic concept. In this case, what's happening is like, you know, we're going to have kids, but I'm going to basically make a bunch of half clones, and I'm going to send all these half clones out throughout the world. Some of them are going to land in really good environments, and they're going to be able to reproduce like gangbusters. So that's a good thing, right? So I'm spreading my DNA to good environments, because mine may not be so great, right? So that's kind of a way to sort of get that spread around into more adaptive and more beneficial environments. So the idea of the sexual life cycle is actually so important that if you take zoology, a lot of times when you go through zoology, you basically study the organisms by phylum, right? That's where taxonomy comes into play. Sometimes by order, but a lot of times by phylum. And the first thing you do, as soon as you come to a new phylum and a new set of organisms, is you sit down and you study their sexual life cycle, right? So you study this. Well, how much time do you spend in the haploid state? How much time do you spend in the diploid state, right? So how do you organize your entire sexual life cycle? Why do you do that? Because remember, the ultimate goal of all biological organisms is to do what? And everything you do as a biological organism supports that goal, right? So basically, before studying an organism, you have to understand that everything that you study about that organism's biology is a big giant arrow pointing directly at reproduction. So the sexual life cycle of an organism is basically the bullseye, the plan for this organism's life. Everything this organism does is pointing at that sexual life cycle, okay, and connected to it. So now that we got ours, we're diploid dominant and haploid not so much. Let's take a look at meiosis itself. So the first thing to understand is meiosis actually has two different phases. There's a meiosis 1 and there's a meiosis 2, and they both have different goals. They're achieving different things. Now, one of the things that we see in meiosis, in the first meiosis, is what's called the reductional division. So the reason why we call it the reductional division is because the goal of meiosis 1 is to separate the homologs. So you're separating moms from dads. This is where you separate mom 1 from dad 1, mom 2 from dad 2. So you're separating the homologs. And when you do that, by the way, you're reducing it, aren't you? Think about it. You normally have 2 of every chromosome. That is to say, your 2N. If you were to separate those 2 homologs from each other, then each pile is going to be half as much, right? So it's going to be N. So at the end of the reductional division, you are haploid. Now in meiosis 2, this is what's called the equational division. Why? Because the goal here is to separate the sisters. Very similar to what we did in mitosis. So here you're going from haploid to haploid. So in the first meiotic division, this is your reductional. So basically you're going to be separating your homologs from each other. So you have two piles, one with each homolog in it, right? Notice the important thing is there's no DNA replication from M1 to M2, right? You've replicated already before this. You're done with replication. You don't replicate again in between it. So this isn't two cell divisions. This is one cell division with two steps. Make sense? So that's the reason why you don't replicate in between. The second one is basically the separation of your sisters. So that you basically, when you take a look at meiosis 1 and 2, it should look something like this. So here's mom 1 that's been duplicated. Here's dad 1 that's been duplicated. The first step is to separate each of these into individual piles. This is M1. And then in M2, you separate the sisters into individual piles with the correct number of chromosomes, right? One chromosome each. Okay. So when you take a look then at the process of meiosis, you can see there's already a lot of overlap with mitosis. So you still have prophase, metaphase, anaphase, telophase. Notice what I mentioned before, last chapter, I said that protometaphase will go away. Notice the absence of prometaphase, right? Is it wrong? No. The events of prometaphase are still occurring. That microtubule fly fishing thing is still happening. It's just that from a meiotic point of view, that's not your focus, right? In meiosis, your focus is on chromosomes and chromosome segregation. That's the reason why we tend not to see a prometaphase when we talk about meiosis. It's still there. The activities are still happening there. Okay. So this is kind of what it looks like. And so one of the things that I want to take a look at is basically where we're going, right? So this is the direction we're going. So here's what your two homologs look like once they're paired up. So you can see you've got your kinetochore attached to the centromere. That's nothing new, right? We saw that in chapter 10, but it's also true for both. So you can see there's a kinetochore hiding under there, underneath there. But now what's happening is your homologs are zippered together with your synaptonemal complex. Okay, wait. Let's back it up here. Back it up. So meiosis 1 and 2 has a prophase. Prophase 1, prophase 2. Metaphase 1, metaphase 2. Anaphase 1, anaphase 2. Telophase 1, telophase 2. So everything is the same, and the events of prometaphase are still occurring too, whether we mention prometaphase separately or not, then you have a valid question that you didn't know you were asking. I'm asking it for you because I know you're going to ask it. Maybe. Is if everything's the same, then what's so special about meiosis? Right? What makes meiosis so special? Well, the specialness of meiosis really comes in prophase. This is basically where you have the innovations of meiosis. Now think about it, and always keep it in front and center in your mind. The purpose of meiosis is to do what? To create the gametes, that's true, but what are you doing when you create it? So think a little more globally, like our first conversation before we got started. The purpose of meiosis, AKA sexual reproduction, is to do what? To reproduce, but you can reproduce asexually, right? So what's the purpose specifically of sexual reproduction and meiosis? Genetic diversity, right? So remember, the kingpin of all of this, the driving motive force, that shining city on a hill that we're looking for, always is genetic diversity, right? Variation, a deep, deep pool, an infinite pool, if you will, of variation. So the question is, how do we get it? By the way, if everything is the same as mitosis, do we have genetic variation? No, so we have to do something different, how we? Okay, so let's take a look at it. The magic happens in prophase. So let's take a look at what's happening in prophase. I'm going to sort of roll forward to our prophase slide. Here we go. So let's take a look at the events of prophase, right? We already know 3 of them, don't we? What are some of the things that happens in prophase? The nuclear envelope breaks down. Yep, that still happens in prophase 1. What else happens? Ooh, 1 out of 3.
Chromosomes become more condensed.
Yes, chromosome condensation. Yep. That's also happening in prophase 1 of meiosis. What's the third one? You have spindle migration, right, into its antipolar position and the development of the spindle. That's all happening here too. So, okay, wait. So it's different. Because that's all happening in mitosis as well. Well, the things that are different, I'm going to list here. First of all, you get homologous pairing in prophase 1. That is to say that the homologs find each other. That there's a mechanism for mom 1 to find dad 1. Okay. Then after you pair, you get synapsis. And then after synapsis, you get recombination. These are all M1 specific. So the homologs find each other and then they synapse together. And then they recombine. Okay. Recombining, by the way, has a more colloquial name, crossing over. So let's take a look at this here for a second. So what does this actually look like? So let's kind of back it up here and just kind of go stepwise through the process. So the first thing you do is the homologs, which is something that doesn't happen in mitosis, right? The mom 1 finds dad 1, and they pair up like a pairing, like a package deal. Mom 1 and dad 1. That doesn't happen in mitosis. In mitosis, mom 1 could care less about dad 1. Mom 1, all the chromosomes are doing is just lining up in single file along the metaphase plate. That's it. Okay. They don't care about finding their partner. In meiosis, you got a mechanism where the partners find each other. That's homologous pairing. Now let's go ahead and send this through replication. So you're going to create now your 2 sisters of mom 1 and the 2 sisters of dad 1. So then we go through what's called synapsis. Synapsis is basically, it's kind of like, remember we had cohesin gluing the two sisters together? If you use that same idea between the homologs, that's like what synapsis is like. Well, in this case, it's a bit more of a robust structure. So there's an entire protein network that basically will grab onto and sort of zipper up the two homologs to each other with some lateral connecting proteins around a central shaft. This whole central structure is what we refer to as the synaptonemal complex. This is that complex of proteins that basically glues the two homologs together so that now the mom one and the dad one are a package deal. So you've tethered them together. Nomenclature. Sometimes you'll read into this. There are two names for this particular stage right here, where you see the two lined up to each other. There's two different names that you'll hear. One name that you'll hear is called the tetrad. Why? Because you have 4 sister chromatids. That's why they call it the tetrad. You'll also sometimes hear this referred to as the bivalent or the bivalent. Bi, two homologs, okay? And so either one is referring to the same structure. And you'll sometimes hear either one of those. So when you hear that, just know that's what they're talking about, okay? So this is before you do anything in alignment. So you haven't even gotten to metaphase yet. This is all the activities of prophase. So homologous pairing, the two chromosomes find each other, and then you zipper them up in synapsis, right? That's what they're trying to show you in this image here. So you can see here you have your two homologs paired together. And then in here you see you've got this lattice work structure of protein holding the two together. That's the synaptonemal complex, okay? And notice there's still cohesin here. But remember, cohesin only holds the sisters together at the centrum. The synaptonemal complex holds the homologs together completely from tip to tip. So it's basically zippering the homologs together, the complete chromosome together, not just tethered at the centromere where they're flopping all over the place. That's what we see in mitosis. This one, they're zippered together completely, all up and down their length. Okay.
So you said that mom one is replication.
This one?
Yeah.
Yeah, so I just put the replication in there just so we can get to this tetrad state, right? Because the tetrad state is post-replication because you've got both of your sisters attached. So yeah, typically, and they don't have, so there's a question as to whether or not the homologous pairing happens during, before, or after replication. It typically happens after replication. I just put it there just for sake of, so you can see both of them, right? Just so that you can kind of see that apart. But they're usually not, they haven't found each other just yet. So they go through replication and then they go through homologous pairing and then all that. So homologous pairing actually happens post-replication. Makes sense? Okay, so then when you cross over, that's the last piece of it, you'll basically create these physical structures called chiasmata. So if you take a look at, two homologs, a chiasma basically will be this X-like structure that connects the two homologs together. Okay, that's what a chiasmata is. It's evidence of a crossing over event between the two homologs. But what I want to do is I actually want to flesh this out a bit. Okay, let's see here. We're going to do recombination. And so what color do I have here? Okay, we'll stick with that. So here's what I'm going to do. So first of all, when we take a look at recombination, because we still haven't answered the first and biggest question, right? Which was, if the whole purpose of this is to generate genetic variation, I'm not seeing it yet, right? Where is that genetic variation coming from? Well, as it turns out in meiosis, there's 3 major big engines of variation. This is the first one. So let's take a look at how you generate genetic variation from recombination. So I'm going to take some chromosomes here. I'm going to take a dad one, and then we're going to take a mom one. So what color should dad be? Let's make dad purple. And so I'm going to draw the chromosome for dad one. Right, so here is chromosome. There's my centromere right there. And there's the other one. Okay. So these are 2 sisters attached to the centromere. That's what that is. Now let's do the same thing for mom. There's mom one. Now, here's basically how recombination begins. Typically, recombination begins by doing a double strand break in an area of the one of the sister chromatids. Okay. And generally speaking, there's an accompanying break in the other homologous sister. Now here's the thing. You can heal this break one of 2 ways. You could either do what's called homologous end joining. You can just basically just reseal the ends, right? In which case you get nothing. Or since both of them are identical, they're the same, homologous, you can actually use the strand of the homologous chromosome, and you can basically connect one end to the homolog of the other. Basically, what you can do is you can connect this purple end to the pink end down here. And you can connect this purple end to the pink end down here. If you do that, that creates what's called a chiasm. Chiasmata for plural. If you do the same thing over here, then you're going to create a chiasma over here. A couple of things about chiasma. First of all, chiasma are randomly occurring. They don't occur at specific places, they occur at random places. So if I do this, if I place this chiasma here 10 times, it's going to be in 10 different places. The number of chiasma is also random, right? So I can have either 1 or 2 or 3. Generally not more than 3, because usually the more you have, like if I get 2 chiasma close to each other, there's a zone of inhibition. We learned about this actually in genetics, in the genetics class, how the chiasma sort of elbow, they kind of throw elbows, right? They're kind of boxing out underneath in the paint. Kind of an idea if you're a basketball person, right? So basically, like one particular chiasma will inhibit this range around it. So the next one can't land right next to it like this. It has to kind of be away from it. It's like a personal space sort of thing. Right. But given enough, big enough chromosome, you can drop in easily 3 different chiasms. So I just added 2 just for sake of illustration. And so what I want to do now is this is something that happens in meiosis. This is what's happening in prophase 1. And a lot of times what we used to think was that recombination was just sort of like a random, like an accident. But it's not. Because what we noticed was that actually recombination is very intentional. As a matter of fact, the lab that I came from, UC Davis and Scott Hawley's lab, who was a big meiosis guy in fruit flies. One of the things that he discovered was that in fruit flies, when you had a strain of fruit flies that were recombination deficient, that is to say, when you repressed recombination in fruit flies, it screwed up their meiosis. So he was actually able to show in his work with fruit flies that recombination was not just a happy accident, right, to go Bob Ross, right, but that recombination was a very intentional requirement in order to get normal meiotic segregation of your chromosomes. And so that's a different ball game now, right? So that basically means that you're dropping in recombination on purpose, right? That's intentional. Notice that also is in line with my standard mantra that random is expensive. And the one thing the cell does not like is expensive, right? So typically when you see things in the cell, there's usually a reason and a purpose and an intention for it, because for it to be random would be very costly, and the cell doesn't have that kind of money. Okay. Here's what I'm going to do. What I'm going to do is I'm going to basically roll this through the entire meiotic process. So just kind of bear with me because I'm going to fast forward through M1 and M2. First, so M1 is basically going to separate the homologs from each other. So we're going to go like this. And then M2 is going to basically separate the sisters from each other. Okay. So that's basically what I'm going to do. And we're going to take a look at what our outcomes are here in our meiotic system. First of all, number one. So we're basically going to take the first one and we're going to start off with purple. We'll pick up the purple centromere and then basically we'll finish off purple. So the first one that we see is going to look something like this. Right. Now let's take a look at the second one. So the second one, we're going to start off purple, but then we're going to, when we hit that chiasma, we cross over the bridge. So we're going to cross over the bridge and then we're going to start picking up the red or pink or whatever color this is. And then when we hit the other chiasma, we're going to go back over the bridge and we're going to pick up the rest of the purple. So what does this one look like? Looks like that. Okay, let's take a look at number 3. Same thing's going on with 3, right? So we're going to start off with the red. We're going to go across the chiasma. Then we're going to pick up purple, pick up that purple centromere. We're going to hit the other chiasma, go across the bridge. Then we're going to pick up that little red tail. So in this case, we look like this. And the fourth one is basically red all the way through. Notice what I have now. I have 4 haploid. Yes. I had 2 before. Now I have 1. All right, 4 haploid, genetically non-identical daughter cells. Didn't we say that's what the products of meiosis were? 4 genetically non-identical haploid cells. So typically speaking, when you take a look at this, you'll notice that some of these kind of look familiar, right? So like this one here looks a little bit like mom one. It actually looks pretty much dead on mom one, right? Number 1 looks like dad one. So 1 and 4 are what we refer to as parental. 2 and 3 are what's referred to as recombinant. A little bit of dad, a little bit of mom. So already you have a shuffling. So for instance, if you were a male, all 4 of these would be moving forward as one potential sperm to use during fertilization. If you're a female, you would basically pick one of these to advance forward to become an egg. How do you select? Selection is random, right? But let's think about, that's a third engine I just mentioned. This is the first engine, a variation. Well, let's take a look at this, right? So you can see basically that the gametes you're producing are not only half of your genome, right? Because they're haploid, but they're also shuffled because of recombination. So recombination will actually shuffle your chromosomes, and at least half of your gametes are the reshuffled type. So that creates a lot of genetic variation, yes? But it's actually worse than you think, or better than you think. Depends how you look at it. Right? How many of you guys think this is impressive for variation? Do you think this is enough variation for us? There's 8 billion of us and going strong, right? Do you think this is enough to basically sort of underpin or explain the mantra of the reason why there's not 2 of you on the planet? And there will never be 2 of you on the planet. Is this enough variation to convince you of that? You know why? Because you are not just the product of your parents' variation. Because when we take a look at meiosis, this is kind of where genetics comes into play, right? Oftentimes what we do is we make the mistake of only looking at just the immediate generations. We look at us as a product of our parents or our kids as a product of us. When in reality, when you look at genetics, that's really not accurate. Because what did your parents have, right? So for instance, when you say, oh, look, half of the time I'm going to look exactly like my parents. So, I mean, how is that enough variation for us, right? Well, wait a minute, wait. Let's actually get an accurate view of what this looks like. This chromosome that you had from your mom that looks spot on like your mom, so you should be like a little mom clone. Let's think about this for a second. That itself is not just your mom clone. Who'd she get that from? She either got it from her mom or her dad, your grandma or grandpa, right? But guess what? That chromosome was likely also reshuffled. So not only do you have grandma and grandpa's genes through mom, but you've got a shuffled version that mom got. So really, when you take a look at your chromosome that you got from mom one, it's really not mom's chromosome, is it? It's a shuffled version of grandma's chromosome, who had a shuffled version of her dad's chromosome, who had a shuffled version of his mom's chromosome. So basically, when you add a shuffling on a shuffling on a shuffling on a shuffling, what you have now is not like, oh, well, my chromosome is just like my mom's. No, it's not. What you have is a chromosome that looks more like a quilt of all these little different bits and pieces that have been shuffled onto that chromosome and off and on and off and on and off throughout the generations. And you can see this, can't you? Because let's take a look at a family, for instance, where you have multiple generations of like especially photographic data. And then somebody marries into the family and adds to the progeny of the family, and they like look very different. Like let's say it's a fair-haired, fair-skinned type of a family, and they marry somebody with darker features or something like that. You can see that shuffling of those darker features as they get shuffled throughout the family and sort of intermixed throughout the family. Right. I mean, initially it's very strong and dominant, but then it kind of gets maybe diluted by a half and sort of shuffled around. So like, for instance, oh, you got the hair of great grandma, but not the eyes, right? So you kind of start to see the separation of some of these individual pieces, right? Hair, eyes, skin, starting to slowly segregate. And so a few generations down, what you have is this patchwork quilt of a chromosome. And some of those pieces you got from that one great grandma who was really dark and had all dark features, right? Which is kind of like an outlier as far as the family's concerned. And so you can kind of see that. So basically our chromosomes are actually like taking a jigsaw puzzle, and then every generation is cutting the jigsaw puzzle in half, gluing it together, and then recutting the jig with the jigsaw, recutting the puzzle. So what you have is a puzzled puzzle. That makes sense? So over time, all you're going to have are like these little bitty specks, but you're going to have like the full puzzle picture. So it's going to get more complicated as you go on because you're going to have smaller chunks because you're going to be swapping stuff on and off. And then this is what's happening just within you. It's happening in your partner as well, right? So they're doing the same thing. So you add a mixed, right? A shuffled deck. And half of that shuffled deck goes forward, and your partner does the same thing, and it's a random selection, right? So basically it makes it nearly impossible to get that same outcome twice. So that makes sense? Is that a question?
I kind of just had a question about like identical twins.
Yeah.
So does that mean
They cheat. No. So genetically identical twins are the same, but actually they're not the same, right? Because even with an identical twin, let's imagine that you had identical twins who are genetically the same. And let's say, for instance, that they had kids with the same person, right? Because then you would have like, okay, well, wouldn't they come out the same? And the answer is no. Why? Because the recombination that's going on in one twin is a random reshuffling of all that genetics. So it's not possible to have the same outcome twice, even in the same person. It's impossible to have the same outcome come out twice in two different people because they are two independent events and they're unassociated with each other. So it would be kind of like this. It's like I said, it's like a random number generator, right? So what is the likelihood of shuffling these numbers around and getting the same sequence of numbers twice from the same number generator? That's pretty rare, isn't it? Because we've never seen the same lottery number come up twice, right? So that tells you there's infinite variation there. But now what happens if you have it happening in two different random number generators? What's the likelihood of being able to get the outcome in A the same twice and the outcome in B the same time twice? So if it's impossible to get it in one event, it's like doubly impossible to get it in both events at the same time. That's what's happening when you are born, right? That's what's happening when you are conceived. The random generator of meiosis, which created the sperm in dad, will never happen again the same way twice. And the random number generator in mom that produced the egg will never happen in mom again the same way twice. That's before you even get to the random fertilization idea, right? So you haven't even gotten to that point yet. So just being able to generate the same sequence, right, from this random shuffling. And by the way, the reason why this doesn't happen is because a lot of times students will mistake and see like, well, half of the time it's going to be the same. Well, no, it's not. You know why? Because when you go to produce the next egg, it's a different random placement of crossovers. So each recombination event will generate a different outcome. Next time, the crossover event may affect this one here. Because one of the things we don't get into, because we get into it a little higher genetics, like advanced care opportunities to get into it. But normally we just sort of show it as these two inner sisters that are involved in the crossover event. In reality, all four of these can be involved in a crossover event. So I can cross over between this one and this one, or this one and this one, or this one and this one. Right? It doesn't matter. So there's a lot of variation. So the next time you go and do meiosis in another cell that might just be adjacent to this one, your placement of chiasma is completely different. The chances that you're going to get the same placement in both of them is nearly zero. Which basically means that these look actually quite different. Some of these will just be one crossover, some will be two. Even if it's two, it'll be differently placed. So you'll have a different set of genes that are associated with the recombinant. And so you have a lot of variation there, right? So basically these are basically an infinite number of shuffle patterns.
So what's up to like in the lab and the parents go and say, I want the green color of the eyes, or I don't know, blonde hair, or
They can't go in and pick. They can analyze, they can like screen for the genes, and they could say like, oh, well, this should have the allele for green hairs. Green hairs are complicated, but they can go in there and say like, well, this individual has two recessive alleles for eye color, so they're probably going to come out with a recessive eye color allele. So they can just do like basic genetics on that one to kind of do that. But generally speaking, that's usually frowned on because that becomes basically eugenics. That's where that goes. Because when an individual, whether they know it or not, the second you start choosing what eye color and skin color and hair color your kids have, you're making a bigger statement than just the look of your kids. You're making a racist statement. That's basically what's happening. That's where that goes. That's the reason why it's frowned upon, because there's really nothing any different than that, than what Hitler was doing in trying to create a master race. That's, I mean, a lot of people would be like, wait, no, that's not what I want to do. No, that's not what you want to do, but that's where it's going. That's what I'm saying. That's where that goes, right? You don't start off annihilating 6 million people in Europe. That's not where you begin, right? The place you began was much more acceptable to German society, apparently. And honestly, American society. A lot of times Americans think like, oh, those terrible Nazis. It's like, go past your history class. We had a pretty robust eugenics movement here in the US too. And actually during the time when the Nazis were sort of revving up and kind of gearing all that up, that was actually in Denver when the KKK largely ran the state. So a lot of times we like to sort of throw shade on other countries that should have known better. We know better than you. It's like, no, you don't. You're Americans. You don't know any better than anybody else, right? You're just more conceited and arrogant about it than the other countries, because the other countries have been around longer than us, and they've fallen flat on their face a lot more times than us. So they know what the taste of mud and blood mixed together tastes like when they do that. America, not so much, right? We still have yet to fall flat on our face and feel what that sensation feels like. So that's my civics opinion there. Take that for whatever you want, right? That was free, by the way. You don't have to agree with any of that. Just if you want to pass the class. I'm just kidding. I'm kidding. Like, he did it. Let's get him. No. So this is just kind of comparing meiosis and mitosis side by side. This is always helpful, right? So here you can see in meiosis and meiosis 1, notice what's happening is you're lining up in double file. And that's important, right? Because what you want to do here is you want to separate your homologs to either side. In metaphase of mitosis, you're lining up in single file, right? Because you're separating the sisters in this particular case. That's it for mitosis, right? But in meiosis, basically you have to do a sort of a similar thing that you do in mitosis, in the sense that you have to resolve it right? So you have to resolve all this stuff. So all this synaptonemal complex stuff that's kind of tethered together. And they actually have the synaptonemal complex kind of in the wrong place here, but you basically have to sort of resolve all this, right? So number one, you got synaptonemal complex holding your homologs together. That has to let go to go through anaphase. And not only that, but you've got your homologs now sort of tangled together with these crossover events. Those have to sort of pull together. So it's kind of pulling a little tangled knotted ball. So you have to get those things resolved, and that has to happen. So this is our crossover. So basically we're taking a look at recombination. And by the way, after taking a look then at the fullness of how that recombination works and how that sort of shuffling or the recutting of the jigsaw puzzle every generation, does that kind of help you feel a little bit better now about how much variation we have? Do you still think it's enough variation to ensure that there will never be another you on the planet? I mean, when you consider the fact that you've never had two lottery numbers the same before, and that's basically what recombination is a random lottery number generator. A lot of times students will be like, okay, yeah, I'm comfortable with that. I mean, I've already have experience with that. We've never had the same lottery number twice. So we're probably never going to have the same shuffling genetic combination twice. Everything is random. Those chiasma are randomly placed. So getting the exact same position of those chiasma twice is highly unlikely. So we're not likely going to get the pattern of the same thing twice. And even if we did get it at the same time twice in one person, we got to get that in the same another person who's completely unrelated. We got to get that all happen at the same time, right? So you're not just asking for lightning in a bottle. You're asking for lightning in a bottle in both of your hands at the same time, right? So the more times you ask for lightning in the bottle, the more ridiculous it should sound. The closer to zero it should sound, right? And that's kind of what we're talking about. And so that's the reason why when you take a look at it, it's intentional, right? I mean, there's randomness, but the randomness is intention. It's intentional randomness, just like our random number generator. It's random because we're intending it to be random, right? So this is a big one, right? So crossing over. And so here's basically your crossover point. And that's kind of what I wanted to do, that separate slide, is to kind of show you what all those, how that whole process works. So that's probably the most and the closest example of how this all works. So you can see here, holding your sisters together, you got your cohesins, and then your synaptonemal complex would basically be in the middle holding your homologs together. And then when you cross over, now you got them further disentangled. And so you got to separate all that stuff. So you got to separate, first of all, your synaptonemal complex you got to break down, and you got to untangle your little crossed over areas. So all of this basically has to happen. So here's our prophase. I think we're going to get too far from this one, right? But this is a nice little review, right? So prophase one, you see the same things happening, right? You got your spindle migration into your antipolar position. You got your nuclear envelope breakdown. And now here you can see you've got your homologs, which have been paired in synapse, and you can see they're crossing over. So you can see like little pieces of them crossing over there. And then you start to move toward metaphase one. Now, metaphase one is the exact same thing, right? So in this particular case, you have your opposite poles, which are attached to each of the homologs. And your homologs, and the key thing, right? I mean, all the metaphase one looks like very much like mitotic metaphase. It looks very similar. You've got the same connections going on. You're attaching to kinetochores. You're attaching to these sorts of things. But the biggest difference is the alignment, where the alignment is side by side. Double file. Why? Because this ensures separation of your homologs. Because you got one homolog on one side and one homolog on the other. So when the two poles pull, you're going to be pulling your two homologs apart. So it kind of looks like this. So basically you have your two homologs that are zippered up next to each other. Got my two poles out here. So when I pull these guys apart, because they're double file, they're going to pull to either side. Then after that, they're going to be in their own cells. Then in meiosis 2, you basically line everybody up in single file, just like mitosis, and you split your sisters apart. So each one of these goes to either side. So the other thing that we also want to kind of show you, so this is what your metaphase looks like. So you can see how you have your metaphase plate going down the middle. You have one set of sisters going to one pole. I mean, so one set of homologs going to the other pole. The other set of homologs going to the adjacent pole or the sibling pole. And that's going to reduce you down from your diploid, which is 2 sets of everything, down to haploid, which is 1 set of everything. And at the end of metaphase 1, you should be happy. Now, what about alignment? Well, alignment is something that we refer to as random assortment. So this, by the way, if you're keeping track, remember I said there's 3 major engines of variation. This is your second. Major engine of variation. So we have the random number generator effect of recombination that's happening, constantly shuffling up your DNA. But now you add to that random assortment. And so what we're going to take a look at, because we're at about our Riching hour, this is a good place to start actually, is we're going to start with random assortment. And we're going to explain how that generates variation on top of what we already have with recombination. Okay. Yeah, I still have to put those up in this week's as well. When I get an opening, which probably won't, the earliest would be tomorrow. Our exam is next Wednesday. And it'll cover probably up and including and completed chapter 11. Is probably what it can, because we'll be able to finish chapter 11 pretty easily there.